Alrighty. Hello, everyone, and welcome back to part 3 of our The Process of Evolution series. I guess is what we're calling it. Our The Process of Evolution series. And, of course, it's me, your favorite Jackson. We also have the wonderful Dapper Dinosaur.
Hello, everyone. I'm Dapper Dinosaur. I'm the most frequent guest on this channel to not be featured in the intro sequence. Fun fact.
Well, Peter, you heard him.
The problem is getting footage of a dinosaur.
It's right there, man.
They didn't have cameras back then, so I think that's the problem.
But there's footage right now. See, it's right there.
We'll have to get that sorted at a later time. I mean, in fairness, we only had PZ on twice, I think. But anyway, hello everyone. Yes, welcome. Okay, so thus far in the series, we have covered most of sort of the bare bones basics of the process of evolution, which is mutations and recombination, natural selection, sexual selection, and genetic drift. So, and then we started speciation last time, which is the process by which one species becomes more than one species. I mean, I guess that's unless you consider
the origin of species.
It is, although I guess it also depends on how you consider chronospecies, how those fit in there.
Maybe we should talk about what chronospecies are, because we have. This is a concept that you get a lot from the fossil record and not so much from extant organisms, because we just haven't been observing them long enough. So a chronospecies is a species that represents, there are a series of chronospecies that represent the same population of organisms that isn't necessarily splitting into multiple species, but is significantly changing its morphology over time to the point that it meets the criteria for a new morphospecies. And morphospecies is something that you get into in paleontology, where you can't use the biological definition with interbreeding populations and things. So you have to say, do these animals or plants or whatever share sufficient morphology that we can reasonably lump them into a species? And yeah, that is arbitrary. And so morphospecies represent a single lineage as it's undergoing morphological change through geological time. So one example is there are 3 named species of the aquatic sloths from South America, but those are all chronospecies. Also, it's very likely that the 2 species of Triceratops horridus and Triceratops prorsus, are chronospecies.
Same for lots of trilobites. There are lots of different groups of trilobites, which you'll have a genus, and it will vary within a lineage, how many pygidial ribs they have over, for 2 million years they'll have 7, then for another 2 million they'll have 8, then they'll go back to 7, and they'll just fluctuate.
For those of you who don't know, the pygidial ribs are a structure of the pygidium, which is the last little bit. Yes. So it involves how many segments are actually merged into that that section of the animal, because a trilobite comes with a cephalon, then it comes with an abdomen, which is completely segmented, and then it has a pygidium, which is sort of like a tail shield. And so the ribs come with each segment that is merged into the pygidium, because I realize that Jackson probably used words that most people aren't familiar with.
Nope, that's a fair point. That's yes. If you guys ever want,
Jackson is too smart for these guys.
I don't know about that, but yeah, if we ever say a word, this is to the chat, that somebody doesn't know, just say, "Hey, define that," and we will be happy to define it.
And I'll try to stay on top of that too, because I feel like I have an okay-ish handle on the kinds of words people know, but probably not.
So yeah. And the reason that there is such a large degree of arbitrariness when it comes to species is species not real.
Yeah,
So
it's
there you
just a
go,
human
people.
category that's handy because, look, we got to be able to talk about things. So you got to come up with words, and you have to put boundaries on what those words refer to.
Mm-hmm.
So, it's sort of like you have to have a convention, but numerous other conventions could work. Sort of like which side of the road do you drive on?
Right.
Left or right isn't the correct answer, but you need to have an answer in order to have a coherent traffic system. It doesn't
Right.
really matter which one you pick.
Right. Exactly. It's a good way to put it. Alrighty. Let's go to the PowerPoint, please. So as I said previously, we started with the concept of speciation, which is when one species becomes two. And typically, if you take a high school biology class or an intro to biology in like college, the typical species concept you'll bump into is called the biological species concept, which was coined by Ernst Mayr in 1942, which basically just says a population which interbreeds and produces fertile offspring is a species.
Mhm.
And as a metric, it's relatively decent. It certainly doesn't cover, and it really just works for sexually reproducing organisms. Obviously, it doesn't work for asexual organisms, doesn't work for extinct organisms. So typically, broadly, for sexually reproducing organisms, this is the species. This is the species concept that just sort of broadly works.
Sexually reproducing organisms.
Yeah, for sexually reproducing organisms. So, as Dapper mentioned, the morpho species concept, you can look at the characteristics that some species in some either living or fossil species has, and compare that with other populations, and figure how similar do they have to be considered the same species, how different, etc. Because you can have variations within species, right? You can have like sexual dimorphism, which we mentioned previously, and so you'll have large morphological differences between like males and females, for instance. Or you may have ecotypes, as we mentioned when we talked about natural selection. So you have populations which vary in their characters by locality. And so are they all the same species? Are they different species? How do you decide? What metric do you use? And so these are all different things that are, or ecology is another one. And so really when researchers are trying to figure out whether or not something's a species, they sort of put all of these things together. What is the genetics? What is the reproductive capability? What is the ecology, the morphology? Is there a fossil record? All that sort of stuff. Those essentially all go in to defining species nowadays because of how much data we have. We have a lot of data, and it has to be accounted for. So,
just watch out for lore.
For lore?
Yeah, Data's evil brother.
Exactly.
It's a Star Trek joke, Jackson.
I got that. Joke. Yep. It took me a second, but I got it. Oh my goodness. Someone mentions, weren't they talking about renaming a bunch of TRex specimens as a different species, but others argue it's sexual dimorphism? That comes from Mackenzie Brown.
Yeah. Who is oddly a notorious lumper.
With TRex?
No, just in general. He's the guy who was like, maybe Velociraptor and Deinonychus are the same genus.
No, absolutely not. But that's a fair point. There was a paper about that. I have not read that paper. I haven't really looked at it. I just saw sort of the press coverage of it. Is it going to be is it going to be accepted by paleontologists? I don't know. Maybe. I mean, it took a while for researchers to figure out that like Stygimoloch and Dracorex were just young Pachycephalosaurus, and that sort of stuff happens when you're dealing with the fossil record, and you have several organisms that look sort of similar, all living in the same area at the same time. There's a possibility there may be some like ontogenetic shifting that's going on in these organisms over time, and we're considering them as like separate species. That happens. It's not unheard of. So
I doubt that it will be broadly accepted, and part of the reason is the arbitrariness of species concepts. The fact that it would for one thing, it would disrupt the literature on the most well-studied Mesozoic theropod ever. Because suddenly you would have all these papers that are using specimens that have always been referred to Tyrannosaurus rex, but now would have to be referred to Tyrannosaurus imperator or Tyrannosaurus regina.
Right.
And I don't think that the minor benefit of being able to sort Tyrannosaurus into 3 morphotypes, which is already done anyway, would really justify that amount of headache, especially given the fact that it's, like we said, it's arbitrary. Plus, as far as I know, all of the known female Tyrannosaurus specimens have been of the I don't remember which morphotype it is, but it's one of them, right? So that's circumstantial evidence leading to the idea that it is sexual dimorphism. And maybe the hypothesis that throughout the history of the species, sexual dimorphism increased. Sure, maybe. I mean, that's an entirely reasonable idea.
Sure, that happens. Yeah.
At most, that means that maybe you name one new chronospecies, not one chronospecies, actually one ancestor species, and then 2 morphospecies. That's probably a bit extreme. That's my take on it.
Like I said, I haven't read the paper. I'll take your word on it. You're the resident theropod, so
it's true.
So when you do get speciation and not when you get cladogenesis, cladogenetic speciation, because that's, I guess, something else we have to talk about. Chronospecies are basically an example of anagenesis. So you have this continuous population that doesn't branch out into more than one species. It's just one species at a time. Cladogenesis is when you have a single population that becomes 2 species. And so when you have that, what you get are sister species. So these are the closest relatives of each other. And here's an example I just picked arbitrarily because we have them at the aquarium and they're adorable. I love Pseudonthis. Pseudonthis tuca and Pseudonthis pascalis. And they're just sister species. They look, as you can see, very, very similar. They both have, this pinkish body or magenta body, this yellow tail. They have these little pointed mouths. They're very, very similar in their overall morphology, and because they probably haven't been separated for very long. There's a little bit of a morphological difference. Tuca has that yellow stripe across the back, which probably helps them with mate identification, so they don't crossbreed. But overall, not a whole lot of differences. Fun fact about Pseudonthis, since I'm talking about them, is they're called Pseudonthis, which means false Anthias, because they were originally members of the genus Anthias, but once genetic testing came out, they were transferred to Pseudanthius, so they're not true Anthias. But then it turned out that Pseudanthius is paraphyletic to a couple other genera, so they're not Pseudanthius either. Or they are, and all this other genera are also Pseudanthius. But it doesn't really matter because all taxonomic ranks are category. You want to have a consistent system, which is why we want taxonomic names to be monophyletic, meaning everybody shares one common ancestor.
And everyone who shares that common ancestor is in the clade or in the taxon.
Right, exactly. And that's for ease of description, because it would be really complicated if, like, we were naming species, but they were related. They were closely related to each other, but in different genera, and it would be very complicated. And so we have this nested hierarchical system, which we will talk more about a little bit later when we get to the evidence for evolution in the past. But
it's almost like ranks are meaningless, and pretending otherwise is a fool's errand.
Almost. This is also one of my favorite quotes from Origin of Species. It actually might be my favorite quote. Chapter 2 of Origin of Species, just read it. If nothing else in the book, just read chapter 2. It's a fantastic chapter. Darwin says, many years ago, when comparing and seeing others compare the birds from the separate islands of the Galapagos archipelago, both one with another and with those from the American mainland, I was much struck how entirely vague and arbitrary is the distinction between species and varieties. And Darwin just spends that whole chapter just bashing the fact that there is no delineation between species, subspecies, varieties, races. He also mentions like neutral theory briefly in a very rudimentary sense. He's like, what if polymorphisms just kind of spread through the population and they were neutral? And that's a really interesting idea for someone who lived like 100 years before neutral theory came out. So, or was formalized, I should say, mathematically formalized. Anyway, read Origin of Species if you haven't done it already. It's a good book.
I second this.
Next slide, please. I do also have to say, don't read it as though you're reading like the most up-to-date work in in evolution. Read it as a historical interest, please, because it's by no means up to date.
No, it is just a like 15, 16 decades or so out of date. I don't know, something like that.
So, it's been a little bit, yeah.
A little bit.
So these are fruit flies from the island of, you're probably going to be better, is Sao Tome?
Oh, that looks like a San Tomea.
Well, that's the...
Oh, wait, no. That's the species name, yeah. Oh, yeah. Oh, because you got to nasalize the like Sao Tome. Look, my Portuguese is terrible.
Yeah, because it's right off the coast of or it's near South America. I should stop saying right off the coast. It's near South America, if I remember correctly. So actually, this is an island where Jerry Coyne did a lot of his research because he's one of the eminent fruit fly specialists, and that's why he became like one of the speciation experts, because he goes to this island which has introduced cobras, interestingly enough, and he studies the fruit flies there, and you have these different
a dumb idea.
Yeah, someone just released cobras on this island. It's freaking wild. Probably by accident, I mean, but still, So so as you can see, the fruit flies are divided into these sort of, I'm not entirely sure if they're ecotypes per se, but they're divided geographically. You have the ones sort of in like the upper part of the mountain of this island, and then the ones in the lower areas near the coast. And they have some slight differences. The ones in the more towards the center, maybe it was to control rats. Maybe, but that seems a bit extreme. Like cats are a thing. I don't know.
Yeah, cats, weasels. There's a lot of things that are way lower down the danger to humans list that kill rats just as effectively as cobras.
So you can see there's some slight morphological differences. The ones near the center of the island are more yellow, and the ones near the coast are more black. This paper is not. I don't think Coyne was a co-author on this paper, but they have Drosophila jacuba, which is the one on the coast, then Drosophila santomea. And so this is also dealing with that biogeography concept that we talked about in the previous episode. So they're adapting to novel localities and having morphological novelties as a result. So, is there an annotated copy of Origin of Species that points out things that are no longer accepted and what the state of science currently is? I mean, you could read like The Greatest Show on Earth by Richard Dawkins, because he does a bit of that. I don't know about an annotated copy of Origin per se.
Not that I'm aware of.
But like, if you're interested in reading about, sort of the history of Darwin as well as the evidence for evolution, Greatest Show on Earth by Richard Dawkins is a pretty good book.
The only thing even close to that I know of for Origin is that weird Ray Comfort edition of Origin that has like that ridiculous intro and cuts out a bunch of chapters.
That he like plagiarized? Yeah.
Yeah.
Oh God, I remember that.
So don't funny. Get one of those. I mean, unless you just want to get it to laugh.
Yeah, I think I have one of Ray Comfort's books around here somewhere, but at any rate. Next slide, please. All right, yet another good reason why species concepts are totally bogus. So, the guys on the left are Insatina salamanders. So the genus and species is called Insatina. I think it's X schultzi. Not entirely sure how it's pronounced. It's got a lot of consonants in it. But basically, these guys are from the Oregon region. They radiated southward towards California, or into California. The only thing is, there's this big desert in the middle of California, and amphibians do not like very hot, dry places typically.
I don't know. I live in Arizona, and we got frogs here, so just saying.
Well, as I was about to say, the ones who do live in very dry areas have pretty specialized adaptations for living there.
The frogs around here will do this thing where they'll coat themselves in mucus that turns into like a dry leathery thing, and then they'll just go into like cryptobiosis for months, if not years.
Yeah, basically, whenever they won't breed until it rains, and then they have very rapidly developing offspring because the puddles dry up in a few days. So, so yeah, you have to develop some pretty
Desert
serious
frogs
adaptations.
are wild.
Yeah, I don't think these guys have anything like that. I think they hug more the forested regions.
That makes sense.
Which is why they're around the desert.
Also, like most salamanders, they're adorable.
They are pretty cute. I don't know if that's like aposematism, or I don't know if they're poisonous. I should say. I do not know with their coloration.
You describe the species complex while I look up whether or not Insatina salamanders are poisonous.
Fair enough. All right, so
but
these
they're
guys
probably not venomous.
Yeah, I doubt they're venomous, but they may be poisonous. Because I know there are Triconutes in California, which are highly poisonous. So, anyways, these guys radiated southward into California, and they largely radiated around this desert. You can see one of the subspecies or whatever sort of cross the desert and end up on the other side. But the cool thing about these guys is on the coastal side, you have the non-splotched, and on the mainland or the inland side, you have the splotched morphs. And so when you do the phylogeny on these guys, you find out, yes, you have like Picta, that guy way at the top, and he's sort of like basally derived with respect to all the rest. And then the unsplotched all cluster together, and the splotched cluster together. And so you have this really interesting evolutionary pattern. But yes, as Origami Swami points out, it is still a salamander. It did not turn into a pineapple. I'd be very impressed if it turned into a pineapple. Also, kind of sad.
Evolution debunked, though. At that point, evolution debunked.
Yeah, no, absolutely. It's like, well, I guess I have to find a new line of work now.
Yeah. Also, they are poisonous. They have poison glands near the base of their tail on either side, apparently.
Aha. Okay.
And they secrete a milky, toxic substance, apparently. So there you go. That's from internets. So take it with a grain of salt. But yeah.
Brainbug is right. We are talking ring species. Yeah, these guys, gals, these animals are examples of ring species. They essentially circle some geographic structure, either because it's too hot, or there's a mountain in the way, or the lake is too deep, or whatever. And so they radiate around the sides of it, and by the time they meet back up, they can no longer interbreed. So once you get to the very southern end of California, the splotched and the unsplotched can no longer interbreed with each other, but they can interbreed with each other to a better extent along the sides. So, and then on the right side, you have the Lake Tanganyika Tropheus cichlids. And so these guys it shows them all sort of along the coast. So I know a lot of cichlids prefer shallower areas rather than deep lakes. So what they probably do is they hug the coast, and they've just sort of spread around and invaded different regions of the coast, which maybe have different like sediment types or come into contact with different environments or something. I think Tanganyika is a pretty long lake. So anything you'd like to add, Dapper?
No, I think we covered it. And yeah, we did find out that Insatina salamanders are poisonous, but not venomous.
Nice. Next slide, please. Just another example of a species complex. This is Amanita Muscaria. Don't eat these, folks. Don't even pick them up. Just don't do it. Just
if you're not an expert on mushrooms and in terms of edibility, just don't eat wild mushrooms. That's my pro tip.
Yeah,
go to the grocery store, get some prepackaged mushrooms.
Right. Yeah. If you can't tell the difference between like morels and truffles and all that stuff, just don't even risk it, because these will kill you. You won't have time to like get your stomach pumped. It will be too late. So anyway, and so the cool thing about Amanita muscaria is these, if I remember correctly, I believe these are predominantly Laurasia in distribution, and so they look like they have these different polymorphisms. So populations, some will look like each other, some will not look like each other, but the thing about it is they're all closely related to each other. They're all considered like the same species, even though they live in wildly different areas, and some populations look fairly different, but they're also considered Amanita muscaria. Yes, the Sisyphean bead points out, well, it is a rift lake. True.
Speaking about Tanganyika, yeah.
Yeah.
Which does tend to make them long and narrow and often deep.
I have, for anyone, what you're probably hearing, that trickling noise is my big Lake Malawi cichlid tank, because I do also love cichlids. I have a whole bunch of them, mostly peacock cichlids.
Do not mix Tanganyika and Malawi cichlids.
Yeah, no, don't mix. Broadly speaking, don't mix the different Rift Lake cichlids. Like Victoria cichlids tend to be really mean. So yeah,
It's just a bad neighborhood in Lake Victoria.
They have cichlids that will pick the scales off of other cichlids. They're like parasites. It's wild. So anyway, anything to add?
I mean, I don't know a whole lot about mushrooms, so no.
Mackenzie Brown says, tank tour. I have a cichlid tank, an axolotl tank. I've had my axolotl for like 5 years, 5 or 6 years, something like that. I've got some Hymenocyrus frogs, and then a big Tetra tank.
This is a great description, but I think we're going to have to see a tour sometime, Jackson.
We'll see.
You'll just have to tidy up first.
That's probably true.
I still want to see a Darwin cam. We need a Darwin
Next slide,
cam.
please. And again, just more examples of species complexes. So on the left, you have some different Himentua. Was it Urnak? Urnak? I have no idea how to pronounce it. It's the like leopard rays, basically. Leopard and reticulated rays. And some are really closely related. Some are not so closely related. They all look very similar. You know, because that's what happens. And then on the other side, you have different tree frogs, which are, you know, Hypsiboas calcaratus, which are fasciatus. Well, that kind of depends. They're very similar morphologically, but they have different calls. One of the things that tends to separate frogs is the pitch of their calls, which is a sexual selection thing. So
some chicks just really dig that falsetto voice.
That's true.
And some are much more into the bass, you know?
Yeah.
It depends.
That's true. Anything you want to add here?
No, I feel like we pretty much covered the concept. We're just giving examples.
Yep. Okay. Yeah, you're right. Basically, species not real, list of examples. There you go, folks. Mm-hmm. Next slide, please. Okay, so the way that I sort of described it earlier was you have a population, and then it splits in two, and you have sister species, or you have your chronospecies, which is just one long lineage. Well, that's not always what happens. Sometimes you can actually get a whole bunch of very close speciation events if a population invades a large enough area. Like on the left there, you have the Galapagos tanagers, and there's, I think, 13 species of them in total. And basically, you had one population who radiated in just 2 to 3 million years on these islands, and they filled a whole bunch of niches. And so it wasn't just one species split into two. It's like a whole bunch of species kind of all at once. And then on the other side, so this is actually a really cool one. On the right, those are the soapberry bugs, because these guys, their beaks elongated when a new fruit was released into their home. So you have the balloon vine, which is not native to the United States. I think it was introduced from China, if I remember correctly. And these guys, these soapberry bugs, they penetrate down into the plant and like whatever it is,
They suck up some juice.
Yeah, they suck up some juice or whatever it is. But when this new plant was introduced, their beak sizes rapidly increased in just a few decades as they started also eating on these balloon vines. So these guys had a whole bunch of morphs, sort of all evolving at once. So, anything you'd like to add?
Bugs are cool.
Bugs are cool. Brain bug, can we get a bug? Bugs are cool in the chat. Also,
Yeah.
if you're reading
Also,
oh, go ahead.
I was gonna say also, I'm still not convinced that bugs as in insects are just like pan crustaceans. Still sounds like crustacea to me.
Look, take it up with the taxonomists. I don't make the rules. I just enforce them.
I'll just say this: are there any crustaceans that are more closely related to insects than to another group of crustaceans?
They're still pan crustacea. That's the monophyletic term.
Well, then crustacea is paraphyletic, and it's not a valid taxon. You gotta pick.
Well, what
you
is
gotta pick a side,
is
man.
it? Crustacea is.
Look, if there are some crustaceans that are more related to insects than they are to another group of crustaceans, then
I agree. Crustacea is not. It's not considered monophyletic anymore. It was changed. It merged with hexapoda and became pancrustacea because because crustacea no longer encompasses.
That's like when people are trying to push Artiodactyla. It's just Artiodactyla, man. You just find out the cetaceans are part of it. I'm done with all this changing names because some group was found to be in it.
It was just one little clade that got moved over. It wasn't like a whole bunch of clades got merged and all that sort of stuff. I don't know. Anyways, apparently Dapper's clearly done with his
taxonomy. I will fight you about taxonomy.
Slide, please. Won't the monophyly mean we are Australopithecines? Yeah, that's a problem with chronospecies. When do you determine it's no longer the original species, or with genera? Because we are direct descendants of Homo ergaster, right? So doesn't that mean
certainly.
doesn't that make us Homo ergaster? Well, yeah, in a sense, it kind of. Doesn't that also make us like? I came across a paper that argued Australopithecus garhi is directly ancestral to Homo. So theoretically, doesn't that also make us Australopithecus garhi? Like, well, yeah, I guess so, because we are direct descendants of them. So,
yeah.
But
so Australopithecus is called an evolutionary grade rather than a clade. A grade is a group of organisms that are united by both their place in time as well as their gross morphology. Mm-hmm. Grades are often be paraphyletic if you were to try to use them as a clade, because they, there are exceptions within that group of organisms taken holistically that are excluded from the grade because they don't meet the morphological idea. One that you'll still hear talked about is pelycosaurs, which are a group of synapsids, and they're the things like Edaphosaurus and Dimetrodon and things like this. And this is not, pelycosaurs aren't a clade because they exclude things like mammals. But they are a grade in the sense that, like, there's a group of these morphologically similar animals that are all living around the same time, geologically speaking. So it's a grade. And it's as long as you acknowledge that a grade is a grade and it isn't a clade, even though unfortunately those words rhyme, which is annoying. Yeah, it is. It's not completely invalid to have evolutionary grades as a thing that you talk about. Unfortunately, when it comes to binomials, though, when you're getting so down into the weeds of like the difference between genera and species in terms of like chronotaxa and whatnot, it becomes virtually impossible to distinguish taxonomic groups like Australopithecus and Homo without blurring the lines between clade and evolutionary grade, which on the one hand is an annoying problem to have to deal with. But on the other hand, it's the problem you want, because it means that there's just so much data that you don't really know how to just toss things into boxes, because there's just so much of a smooth gradient between the groups. So,
right, exactly, yeah. and another slide for cichlids because I love cichlids. So this is a phylogeny of haplochromine cichlids. Well, the one on the left is the phylogeny. I'll explain the one on the right in a moment, because it's not technically a phylogeny. So what happened? See, what had happened is the haplochromine cichlid. It's called the haplochromine superflock because are they all members of Haplochromous? Well, no, not really, but they're termed the haplochromine superflock because a lot of them are haplochromous in Lake Victoria. Again, it's arbitrary, but where they came from was Lake Kivu, which is a much smaller and older lake than Victoria is. So even though Victoria has way more species, they're all really, really closely related to each other, as you can see by how short those lines are. They're really, really short. That means that the speciation events occurred really recently, and in terms of like 100,000 years or so, that's really recent in evolutionary terms.
Yes, that's basically just now.
Yeah, it's super recent. And so the cool thing, we did a video on this channel a while back titled "An Analogy for Human Dispersal," and the point of that video was there are some people who seem to think that humans originated in the Middle East. The only problem is there's no evidence to back that up.
In fact, there's evidence to disconfirm that.
Yeah, specifically the argument from genetics, which is if you do a phylogeny of all humans, you find out that indigenous African populations are paraphyletic to the rest of humanity. All Africans form a clade together, and then there are multiple clades of Africans. Now, what that means is we couldn't have originated outside of Africa. It's just not possible. That's what that means. But
I mean, the only possible exception would be if the entire population of humanity en masse just decided to move to Africa out of wherever else they were and then spread out from Africa again, which is not what you'd call parsimonious. Let's just say that.
No, that
seems extremely unlikely.
Bonkers.
Right.
Yeah, no, it didn't happen. It's not
Excellent. So, I have to correct you a little because you said they're more closely related because of the shorter lines. If Kent Hovind's going to watch this, he's going to have a field day with you on Whack an Atheist. And right after that, you said we have multiple clades of Africans. He probably will call that racist.
I mean,
you have to be careful.
He's a convicted spouse abuser, so who cares about his opinion?
Yeah, I mean, they're not. Well, they're not. It's not recent because the lines are short. The lines are representative of how recent their split was. And so the cool thing about it is the African populations, the indigenous African populations, are more genetically separate from each other than I am, for instance, from a Chinese person. They're more separate from each other than that. And so you can do, and so again, that's another independent line of confirmation. So you can do the phylogeny, you can look at the genetic differences and the similarities, and those are two independent lines of confirmation in addition to the fossils, of course. The oldest human fossils come from Africa.
There's also another bit that's not just looking at the various clades formed, which is that African populations have much higher degrees of linkage disequilibrium, which is to say that their chromosomes are better shuffled, essentially, because every time you do
linkage equilibrium, right?
Well, they have more linkage. I think it's disequilibrium is more shuffled because equilibrium means you have a good ability to predict what alleles are associated with each other. So what happens is when you have meiosis going on, right, chromosomes will cross over a little bit. So they'll say, you have two chromosome 1s, and each one has its own set of genes. Well, during meiosis, those chromosomes will link up and they'll swap a bit. And this happens a few times, usually every time there's meiosis in a cell. So this is producing your gametes. Well, with a population that's been long-standing with a lot of interbreeding for a very long time, you have a long history of these crossover events on all the chromosomes. So which alleles are associated with which other alleles by being on the same chromosome gets very well shuffled up. Whereas with smaller groups who have had fewer recombination events throughout the history of the founding of that group, you can find more closely related alleles in terms of the frequency of finding them together on the same chromosome.
Right.
And that's because those populations haven't existed for as long.
Right.
And that also, so linkage disequilibrium also increases as you get closer and closer to Africa, and it decreases as you get farther and farther away.
Yeah, almost like humans originated in Africa. It's almost like we have a consilience of independent data points, all indicating that humans originated in Africa.
Yeah, it's kind of like going with any other option is completely insane or dishonest.
Yeah, or both.
Yeah, that's an option.
And so the cool thing, sort of relating that to the cichlids, is if you think of the Lake Victoria superflock as like all of the non-Africans, sure, there are way more like groups, quote, quote, of non-Africans, but they're all more closely related to each other than the different African populations are to each other. That sort of stuff. And so you can do the same thing with the cichlids. So the cichlids of Lake Kivu, even though there are a lot fewer species, they are more different from each other than the Lake Victoria cichlids are from each other, or are to any of them. So you can apply the same sort of logic to humans or cichlids, and you get the same pattern. Because if you want to deny that for humans, you also have to deny it for a lot of other organisms, and that kind of screws a whole lot of stuff up.
Or you because it's almost like
yeah, I guess you could.
There are people who are perfectly happy to just be inconsistent. I
guess. The other picture is those are alleles, not species, and so you have this sort of phylogeographic distribution of these alleles. So it's like which alleles end up in which lakes, and how are these alleles related to each other. And so basically for each of those little dots, that's like a different mutation between the different alleles. And it's a really cool chart, in my opinion. So like you have that one big circle on the left, which has a bunch of colors in it. So like that particular allele is in a bunch of fish in a bunch of the different lakes. And then, but you can see most of them are like restricted to particular lakes. It's just a really cool chart. I highly recommend that paper for Hayen et al. 2003. So, next slide, please. Don't talk about the bull sharks. Are there bull sharks in Lake Victoria? I don't think there are bull sharks in Lake Victoria. I don't know what you're talking about, Brainbug. Fossil speciation. Okay, so again with the chronospecies. So, well, this isn't quite a chronospecies, but with certain organisms, we have amazingly good fossil records, just absolutely astoundingly good fossil records, and that is especially the case for what are called phytoplankton. So basically, little photosynthetic protists, essentially, quote quote, because that's also a grade.
Yeah, protist isn't a real thing. It's a grade.
Yeah, protist, not a real thing. And these guys, they make these tough, either internal or external shells, or tests is what is the technical name for them. And then when they die, the test, which is like calcium carbonate or silicon or what have you, precipitates to the bottom of the body of water, and that becomes part of the sediment. And so over millions of years, you form this just absolutely amazing record of these phytoplankton, whether foraminifera, diatoms, coccolithophores, just huge, crazy good records. And so here's an example. These are meogypsinid foraminifera. And so you can see this gradual change as you go up through the geologic column. It's a really, really cool chart. I'm a big fan of this chart. And yes, RJ and I discussed index fossils in the rocks were there. So if you don't have that book, why not?
Mackenzie Brown knew protists couldn't be real. She knew they were that she's been mad about protists since learning that kelp is a protist. Yeah, protist is a nonsense thing. it basically just kind of means single-celled, probably eukaryote that isn't an animal, plant, or fungus.
Mm-hmm.
That's all it means.
Yeah, basically. A brain bug thought we were talking about Lake Victoria, Australia. Nope, sorry, we were talking about Africa. Lake Victoria,
Yes.
Africa. Kivu, Tanganyika, Malawi, those are all the African
I
lakes.
couldn't even imagine a bull shark getting in there, like just being dropped in by a human.
Yeah, that's why I was kind of confused about that. But regardless, next slide, please. So here's another example. This is an interesting paper because this is back when Gould and Eldridge were arguing that punctuated equilibrium happens. And Philip Gingrich, who is a big name in the whale evolution circles, if you've done any reading of whale evolution, you probably come across him and Thuisen, I think, is the other big guy. Then you'll come across their name. But Gingrich was also very opposed to punctuated equilibrium back in the 80s. I don't know if he's changed his position now or if he's even still alive. I have no idea. Regardless, this paper that he wrote, or that he published, kelp aren't single-celled. Technically, no, they're a member of the phaeophytes, the brown algae. They're in the SAR group, or SAR, the chromista. Regardless. Anyway, and so he publishes this paper, and he's like, if we have a good enough picture of the evolution of any particular group, you can see that it's always gradual evolution, which technically isn't what Gould was arguing against. Punctuated equilibrium includes gradualism. He does not say gradualism is not a thing, and that was also
people were sort of straw manning him with that because he was pushing big for the whole relatively rapid morphological change and certain circumstances, and you get us versus them, and then you just make an extreme version of the them guys. So it happens in science too.
Right. In the last episode, we talked about the selectionist versus neutralist debate, which was not supposed to be a debate at all. It was just people misunderstanding how neutral theory works.
It was supposed to be yes and, not either or.
Yes, exactly. Yeah. So those little dots, you got Hyopsodus and Hapomylus, and you can see how there is this very gradual morphological change as you go up through the geologic column. This is the Green River Formation over the course of 3 million years. So again, really cool. That's really, really cool. It's too bad there are people who think that the Earth is 10,000 years old. Sorry.
Yeah, and this all happened over the course of 30 years? Something like that?
Something like that. Yeah, no, that's just bonkers. That can't have happened. So
yeah.
Literally not possible. Next slide, please. Just another example. This is a sea urchin micraster. In case you guys didn't know, sand dollars are sea urchins gone flat. Fun fact for you.
It's true. That's why they're pentaradial.
Yep. So here's just an example as you go up through the geologic column. I think that's the late Cretaceous. Yeah, Setonian, Campanian. Yeah, that's late Cretaceous. As you're going further up the column, you see this gradual change. This is paramorphocline, so they're extending the development of the adult. So, just another example. Thought you guys might like. Next slide, please. Yes, hybridization. So speciation isn't always about one species becoming two. Sometimes you can have two species who become one. Now, in this example, that's not what happened. This is a monstrous hybrid between paddlefish and sturgeon, which I don't even know how this happened. This is just crazy. But I guess according to the kind, that makes paddlefish and sturgeon a kind, I guess.
Yeah, even though their divergence time in real science is isn't it over 100 million years ago or something crazy like that?
Yeah, it's at least 100 million years. It is a very long time.
Completely insane that this is a hybrid that happened, but whatever.
I read a paper on sunfish, which is Centrarchidae, not too long ago. The freshwater North American sunfish, not the mola mola, which is out in the Indo-Pacific. But what the researchers were looking at was how over the course of about 30 million years, which is about how long this group has existed, the further you go back in time with the common ancestors, the less likely hybrids are to happen. That's over 30 million years. And basically, by the time you get to your most separated clades, they're just not having hybrid offspring anymore. This is 3 times that, at least.
Is that why elephants and pine trees can't have babies?
Something like that. I mean, they've
Okay.
only been separated for what, like 2 billion years, something like that.
So around that, yeah.
Yeah, absolutely crazy. If you guys want to, yeah, I mean, it's just bonkers. Absolutely bonkers. Paddlefish are really cool, though. Highly recommend reading about them. They're neat fish.
I mean, sturgeons are okay too.
Yeah, if you like caviar, I guess they're fine.
I mean, I kind of do. Not that I eat a lot of caviar. Jackson's not paying me enough for that.
Or elitist dinosaur over here. No.
What? I mean, I have had it. It is tasty. I'm not going to pretend
Is
it
it?
isn't. Yeah,
I've never tried it.
It's salty and it's a little on the fishy side, but it's fairly mild, and it has a little burst of that salty flavor when you bite into it because the eggs pop, and
Yeah.
so you put in a little like cracker or something, you just eat it. Don't like just spoon it into your mouth though, because that's too salty, too fishy. It's a
Yeah,
it's a little
that sounds
tiny
kinda
thing.
nasty.
You put a little tiny bit on like a cracker and you eat it with a
Yeah.
bone spoon, not a silver or steel
A
spoon.
bone spoon.
Yeah, so the reason is that the metals will leach into the relatively acidic eggs, and then your caviar will taste like metal. So you want to use wood or bone.
Very interesting. The things you learn with Dapper Dinosaur.
Who, again, does not have a whole lot of caviar.
Next
I think
slide,
it's
please.
been a decade.
That's a little bit.
Yeah.
Next slide, please. There we go. Okay. So this is an example in which we have a new species that arose from some other pre-existing species. So we have the largemouth buffalo, the smallmouth buffalo, and these guys have interbred so often they've actually produced a hybrid species called the black buffalo. We actually have smallmouth and black buffalo at the Shreveport Aquarium. So come by and check them out if you want to do that, if you want to see them. But this can happen. This is what's called a reticulation. So you get 2 species which interbreed, and of course these 2 species still exist. You can still find largemouth and smallmouth buffalo. They're just they're still doing their own stuff, but it just so happens that they've also interbred enough to produce the black buffalo, which now can interbreed and go on their own merry way.
I'm a little disappointed that these aren't mammals in Bovidae. Just saying.
I had no idea that people called these buffalo. I thought, like, I was working at the aquarium for a little bit before I realized this is what people were referring to. I thought they were talking about, like, because they're carp. These guys are carp. And so I was like, buffalo? What are you talking about? Anyway. Next
They
slide,
don't even
please.
have horns or hooves.
Nope.
You would get so little gelatin out of those.
They're hoofless. Hoofless buffalo. It's behooved that they are without horns.
Ooh.
Sorry, folks. I'll fire myself. All right, angelfish.
I'm now taking over Jackson's channel.
That's fine.
This is now Dapper Dino 2.
You'll make videos more often than I do. I guarantee that. So that's fine.
It would be hard not to, right?
At this point, yeah. You have these are different. I was going to say cichlids. These are different angelfish. So the first 2 columns are like separate species of angelfish, and then the third column are their hybrids. So like the top column is the Griffiths angelfish and the goldflake angelfish, and that produces that little hybrid. It says sympatric hybridization. I don't remember offhand if the hybrids are also their own species. I don't remember. I just don't know how often their hybridization is. So, sorry, folks. But this is just another example. Hybridization happens because even fish agree species not real. so there you go. Even fish agree. Anything you want to add?
No, I think this hybrids happen, guys. It's a little odd we haven't got a mule yet, as is the classic example of the sterile hybrid.
I didn't even think about that. Yeah. So there's this rule in there is someone who thinks that caterpillars are velvet worm and insect hybrids. I remember reading about that, and
Wait, what?
Can't remember who said it. Yeah,
that sounds like a David Peters nonsense.
It wasn't him; it was somebody else. But yes, who claimed butterflies are the result of velvet worms and insects hybridizing, which also that's a Cambrian. That's 500 million years ago. Is when these guys separate. At least, probably more than that. So, yeah, I don't know about that, but yeah. So the thing about so there's a rule. I think it's called Haldane's rule. It's basically that you can have hybrids, but only if it's the one with two of the same chromosome. So, if it's like a female offspring of a horse and a donkey, then it will be fertile. But if it's a male, the mule,
it
will not be.
Can be fertile. It usually still isn't.
Right. It has a slightly higher chance of being fertile, I guess I should say.
Like the historical instances of confirmed pregnancies coming to term in a female mule can be counted without running out of fingers for a human. So it's not impossible, but don't plan on getting yourself like a mule true breeding farm going where you just produce mules from other mules. It's extremely unlikely.
Right. Yeah. Next slide, please. Because I don't remember. Okay, and then there's just
turtles.
This is just another example. Well, this isn't. I mean, sort of. So basically, these are all the same species, in my opinion. They're all Trachomys scripta. They can all hybridize. They just have slightly different morphologies. I don't think I included a slide on prezygotic and postzygotic mechanisms of isolation, but to give you sort of a prezygotic means they are mechanisms of essentially preventing pregnancies. The mechanism of preventing pregnancy. So before the zygote forms, and there are lots and lots of different ways that can happen. There are different, you can have different behavioral strategies between different species. Maybe one species mates at dawn, the other at dusk. There are morphological isolation, behavioral, all sorts of stuff. So those are.
Brainbug asks which one is Leonardo, and it's actually the Western red-eared slider at the bottom, because canonically the Ninja Turtles are in fact Western red-eared sliders.
Nice.
Yeah.
Zona pellucida,
which also then caused an increase in the popularity of the Western red-eared slider in the pet trade, which has also resulted in it becoming an invasive species across large swaths of the world. So
I believe that.
Not as nice.
There's a like a tissue around the egg, if I remember correctly, and that's the zona pellucida. And it basically, if you're too distinct genetically, the sperm can't penetrate that and then deliver its necessary genetic material. Fun, absolutely wild fact, and RJ and I talked about this in the rocks were there. If you remove the zona pellucida from hamster eggs, humans can fertilize them. So, that happens. That's actually a way that sperm viability is tested in humans. That's why it's done. That's why we know that's a thing. So
fun times.
Yeah, fun times. That's where that goes. Post-zygotic, even if a pregnancy happens, as Dapper said, it may not come to term. It just the parents are too genetically different, and after it starts trying to get the proteins going and everything, you're trying to transcribe genes to make proteins, all this stuff, and it just fails essentially. The chromosomes are too different. Maybe you have aneuploidy, or something like that, and one parent has a lot more than the other, and so he just doesn't work out. That's post-zygotic.
My brain is still on the first fact. So humans can fertilize hamster eggs.
Yes,
if you remove the zona pellucida.
If you remove the zona
let's say
we can't do it normally.
Let's say we take a random young Earth creationist and have him do that, we can have a Ken hamster in a wheel. I want to see that.
I'm going to go out on a limb and say that I'm pretty sure the resulting zygote is going to fail to develop into a viable, like, two-term organism.
There's a good case to be made. There's a good case to be made that goes for all young Earth creationists.
The Ken
well,
hamster,
except there, it is pretty fun. But there are, living and even reproducing adult young Earth creationists out there. So,
the thing about actually, I didn't even know that until Georgia Purdom brought it up, who is a young Earth creationist. For anyone who's not familiar with,
weird way to learn that fact.
Funny how it led me to Ken hamster.
Right. So she and Bodhi Hodge wrote this article about like, what is a kind? Well, we're going to tell you, except we're not going to tell you. We're going to kind of dance around it for an article and then pretend that we answered the question.
They never
as
tell you.
Creationists want to do. And so in this article, so she mentions how creationists have, and I'm sorry, side tangent people, but creationists have this whole concept about like a family is about the level of kind, because broadly, well, in some cases you can get like or you can get a hybrids between different species in the same family, but then I guess it was pointed out to them at some point, yeah, but we can fertilize hamster eggs, and they're like, okay, but it only goes so far in development, it doesn't go all the way. And it's like, yeah, but it's kind of arbitrary endpoint.
Still hybridizing.
Yeah, it's still hybridizing, at least for a little bit. And as I said, they dance around it and never really give you an answer because there's no such thing as a kind. So
yeah. Nor is there such thing as a family, by the way.
No, there isn't. Exactly right.
As we talked about, the ranks that we like to give certain taxa are completely arbitrary. Like the family, like, I don't know, Canidae isn't the same kind of taxon as, say, like Varanidae, which is the monitor lizards, right? Which one of those taxa is bigger? Which one was founded earlier? Like, there's no way to compare them. The fact that they both end in IDAE is just an artifact of human convention. And so that, kinds are at about the family level. There's no such thing as the family level.
And here we have Dapper Dino agreeing with Kent Hovind. There's no such thing as a family. Sorry.
Not in his house, anyways. Oops, sorry.
Yeah, that's more about him, body slamming his partners and less about biology.
Yeah. Anyway, next slide, please. Okay, phyletic gradualism versus punctuated equilibrium. I probably shouldn't have said versus. I should have said and, but regardless. So,
well, you're contrasting them, so it still works.
Sure, yeah. They're being contrasted. These are not opposed to each other. They both occur. Mm-hmm. There's this major misconception that phyletic gradualism was like around and then it was replaced with punctuated equilibrium. No, they work together to both explain how biodiversity changes through time. That's how that works. So phyletic gradualism states that populations are constantly slowly evolving, slowly splitting, and then punctuated equilibrium states that populations undergo short periods of generally allopatric, that means they're separated by geography, evolution, followed by long periods of morphological stasis. And as I said, PE did not replace PG. Again, there's a bit of a problem with what does morphological stasis mean? How do you quantify that? While you technically can, like, there's no 1-to-1 ratio between like different clades and how much morphological change is achieved. Because again, these are human constructs. We're just trying to apply these ideas to help us understand, albeit imperfectly, how these organisms have changed through time. So how much, to go to the example Dapper gave, how much have varanids changed versus how much have canids changed? Right? There's no way to just put them 1-to-1 and compare them. You can't do it.
They're too distant. Like, maybe you could do like genera within that. You might be able to say like, how much has Canis changed versus like Vulpes? Maybe you could do something like that, but even that's on pretty shaky ground.
Mm-hmm. Yeah. Yeah. So it just means, how much there's organisms may change by what we consider a lot morphologically in a geologically short amount of time, because we're still talking hundreds of thousands to millions of years. This isn't overnight. So anything you want to add to this?
I mean, the really big thing that I do think it's important to hammer home is that even in cases of punctuated equilibrium, we're still not talking about like a saltational thing, right? So
right.
You see, if you were to watch it in real time, you're still going to see a gradual change in morphology. It's just that it's going to be significantly rapid. So this is on a spectrum.
Mm-hmm.
So on the one side, you have very gradual morphological change. On the other side, you have relatively rapid change. And in the middle, there's a whole lot of room. Right. So that's the one big thing there is, this is a spectrum. It's not two discrete things like, oh, the species just switched into punctuated equilibrium mode. No.
Right. Saltational changes are much more rapid.
But also saltationism...
Oh, sorry. Go ahead.
I was going to say, saltationism is an idea that was proposed at various times. And there are some changes that we have observed that you could say are kind of saltational.
Right,
but
like
basically the idea is... Essentially. Right. The idea is that you have a significant, like very obvious in a single generation morphological or genetic change, like polyploidy or, a radical redesign of like limb structure or something. And those aren't impossible, but they almost never happen. And also it's often hard for that new creature to then backbreed with the original population,
right?
Which can cause problems for the idea of this continuing on. So, for instance, if you have a polyploid event in a plant, unless it happens to come across another plant that's also had the same polyploid event, it's unlikely to found a new species. Now, that can and does happen, but it's not every single time you have an event of a whole genome duplication in a plant is that going to be a result in a new species, because chances are, most of the time, it won't find a mate.
Right. Right. Exactly. Okay. Yeah, I think that's pretty good. Next slide, please. Living fossil is a term. Actually, Darwin came up with this term. I really dislike this term, but and this sort of plays into the what we're talking about with punctuated equilibrium. So you'll have some group, some clade, which changes quote little morphologically over a long period of time. What does that mean? What does it mean for a clade to change little morphologically, especially when we can't see the genetics going on? How do we quantify little morphologic change, right?
Like, so I think the important thing is it's an informal term,
right? There is a term called stabilimorph, which is sort of an attempt at a more at a slightly more objective framing of this issue. Stabilimorph is defined as a genus that lasts longer than one extinction event, at one major extinction event. So basically, any genus that has been around since at least the end of the dinosaurs is considered a living fossil. But technically, by that definition, coelacanths are not living fossils. The modern coelacanth, Latimeria.
Well, they might not be, because we don't actually have a fossil record for Latimeria, so who knows how long they've been around.
But it's generally considered like at the max 60 million years, isn't it? Which still postdates the...
It does, but I'm just saying it's still at least a remote possibility that Latimeria might be older than we think, because again, we just don't really have a fossil record for them.
Yeah. If you live in the deep sea, typically you don't have very good fossil records. A lot of the coelacanths in the record are freshwater. Or brackish, because, or at the very least near coast, because those are the places that tend to get better fossilization than the deep sea. So, it's useful for lay people. You're right, Mackenzie Brown. Limulus, the horseshoe crab, has been around for like 90 million years, I think. So that is a stabilimorph. Paddlefish, I don't actually remember what is the modern paddlefish. I can't. Oh, Polyodon. I actually don't know if Polyodon predates the Cretaceous extinction.
I believe garfish count as staphylomeres, but I would have to double check because
That's Leptosostius, right?
I don't honestly remember, but I do know that if you look at like Mesozoic garfish, they're, other than changes in size, they're very hard to distinguish from modern garfish.
Right, and I think there are also Mesozoic bowfins, which are Amaya, and they're pretty similar overall.
Ooh, fun fact: garfish means spearfish because gar is the Old English term for spear, and it's the only place where that word survives into modern English.
Yeah,
And they're called that because they kind of look like spears.
They do kind of look like spears. Kappa 1611 says, I prefer living fossils. Staphylomeres trying too hard. Who cares how many extinction events? It's just attempting to add a bit of objectivity to the definition. That's all it's trying to do.
Science likes to be able to say for certain whether something is a thing or a different thing. Yeah. And with living fossil, it's just sort of like eyeballing it. Like, yeah, this looks a lot like its ancient ancestors or ancient relatives. So we'll just kind of call it a living fossil. And like, I mean, okay, are shrews living fossils?
Right.
Because we have shrew-like organisms since like basically since the Jurassic.
Right. Yeah, absolutely.
So maybe, I don't know.
Next slide, please. And then I think this is the last example. Horseshoe crabs and relatives. So essentially the modern, quote, horseshoe crab form, Limulus, has been around since like the Jurassic. Okay, so I thought that was Mesolimulus. Okay, maybe they do go back to the Jurassic. Anyway, as you can see, they've been around for a long time. A pretty darn long time. There are only like 4 species of modern horseshoe crabs, and I think they all split about 70 million years ago, something like that. So that they're probably like chronospecies. Who knows?
One fun thing with horseshoe crabs, which is not related so much to evolution as it is to just cool stuff. Their hemolymph is 1, a really cool, bright, vibrant blue color, and 2, it's actually commercially harvested in order to provide medical treatments because they have a very unusual immune system compared to, say, tetrapods, where rather than like attacking and just individually targeting bacterial cells with various kinds of immune cells, they actually have proteins that clump around foreign bacteria and simply prevent them from actually moving around the body until they die. And so this can be used to diagnose bacterial infection in otherwise hard to diagnose cases because you can introduce samples from the potentially infected person to these proteins. And if the proteins flocculate around something, that means that there is the presence of some kind of foreign bacteria. And right now there's actually a controversy because Limulus is not exactly the most numerous of animals.
Yeah, it's endangered, I think.
Right.
Or at the very least vulnerable.
So the thing is, the blood harvesting is technically non-fatal, but there have been studies suggesting that a significant fraction of those horseshoe crabs that are harvested for their hemolymph do not survive until the next breeding cycle, despite the fact that they're not old enough to have likely died before then. Yeah. So while it's technically non-fatal, it does seem like it's a pretty severe detriment to the overall health of the animal. So there's a lot of controversy about like how should that be regulated. Should we look into some kind of way to like farm a subpopulation of horseshoe crabs for medical use? I mean, people have done it with other animals. There are now medical leeches that are kept in sterile conditions specifically for blood draining purposes. They don't mix with the wild populations. So maybe.
Yeah, absolutely. Definitely possible. Something to look into. All right, next slide, please.
Also, they're monsters when you flip them over.
They do look scary, but at the same time, you know, they're just big babies.
They're not dangerous.
They don't really
but you flip them over and you're just like, oh my god. So
many legs.
So many.
Plus they're chelicerae or pinchers, so that's, you know.
They have chelicerae.
That's what I just said.
Yeah, well,
chelicerae or pinchers.
Yeah, but that's chelicerae. That's why they're called that in the first place. It's because that's the primitive condition.
Right. Yeah. The what spiders have, where it's a fang, that is a derived form, and sulfugids. They also have the big pinchers, which are scary.
They look scary. Look,
but they're not venomous.
Yeah, I found one in my bathtub not that long ago. I just brought them outside.
I mean, yeah, they're too close to looking like spiders for me. Like, I can handle scorpions. I can handle opiliones, which also have the
chelicerae
grabby claws rather than Yeah.
What about Amblipigi?
I can handle Amblipigi. I can handle Europigi. Basically,
Really? Amblipigi have the most horrifying pedipalps in the world.
I think they're adorable. I can handle every arachnid except for spiders, so Aranea, and Sulfugids, because they look too much like spiders, even though they're not closely related. So,
okay.
I mean, as distant as like 2 orders, maybe. So,
what about mites and ticks?
That's what Meristomata. I mean, does anyone like mites and ticks? Who's not like a mite and tick researcher?
I mean, I don't have a big problem with mites. I'm not a big fan of ticks, though. Mites are fine.
I guess it depends on the mite.
As a group, I don't have a problem with mites.
Are we talking about Vinegaroons? Yeah, Vinegaroons are the Europigids. Amblypigids are the tailless whip scorpions, which I think are adorable. If you guys know Just a Walking Fish, he has a tailless whip scorpion.
Nice.
It's pretty cool.
I saw a horror movie where tailless whip scorpions were the ultimate monster, and it just deflated any sense of tension because I know exactly how harmless they are.
Yeah, okay. If any of you guys have seen, what was it, Fear Factor, like back in the day, there was one of the challenges was they tell this woman, we'll give you like $100,000 if you eat, what do they call it, a giant cave spider. And they bring her over to this roulette wheel that has a bunch of Amblypigids on it. And I see that, well, I mean, back then I was like, oh, that's horrible. That's gross. I can't believe she did that. But I saw it recently. And as soon as they like brought her around and showed her that they were Amblypigids, I was like, oh God, no. They're totally harmless. They can't defend themselves.
Yeah.
And she did it. She just stuffed one in her mouth and ate it. And it was horrifying.
Not a big fan of that show for reasons like that. Just mercilessly slaughtering random, scary looking but inoffensive animals is not okay in my book.
Yeah, I mean, I'm sure she didn't know. She probably thought like, oh God, this isn't, a spider. And the layperson probably wouldn't know or wouldn't stick around long enough to figure it out.
There's a reason that one of those was the basis for the most deadly spider in the world in the Harry Potter movie. It's because they look scary.
Yeah, and there's that really stupid line, Daddy Longlegs are the world's deadliest spider. They just can't they can't bite because their mouths are too small. Yeah, no,
turns out
totally.
Both of those are wrong.
Every bit about that is completely wrong. They're not spiders. They're Opiliones, not Aranea. They don't have fangs. They don't have venom glands. So nothing about that is right.
But their calystrae actually can break human skin. They just tend not to try.
Yeah.
So plus, literally every single aspect. Yeah, every single aspect of that entire story is completely wrong.
Yeah. Yeah.
But taxonomy.
Yes. Okay, so essentially we have now covered the process of evolution. Now I want to talk about peacock spiders are cute.
They are.
The harvestman. I meant the harvestman. That's yes, brain bug. We know there are lots of things called daddy long legs. So now we're moving on from the process of evolution, and now we're going to talk about the evidence for evolution. Does that sound good, Dapper?
I mean, I feel like I've heard that there isn't any, so let's see what happens.
So the first bit that I chose as evidence of evolution is the fact that at least among all like the higher taxonomic ranks, like plants, fungi, animals, things that don't do horizontal gene transfer regularly, they fall into what are called nested hierarchies. So essentially, you have a group of organisms. You have a twin nested hierarchy. So you can go in one of two directions. And so with this, it goes from like all animals to Canis lupus. And I guess they consider regular dogs Canis domesticus. So that's fine. But so you have.
I'm not fine with it, but I'll let it slide this time.
What is
Canis
a species?
lupus
Anyways.
Domesticus.
So what they decide to do. So what you can basically do is you can go down one of two branches, right? Because after a speciation event, you have two new species, right? You had one species, which is now two. And from that, each of those species can then theoretically go on to produce two species. And then each of those can go on to produce two, and so on and so on and so on. Now that's Of course, it's theoretical. That's not always how it's going to happen, but that's an idea. That's one way that it can happen.
This is somewhat idealized.
Yes, and so what that produces is you have nests within nests, right? You have these two species, and they're more closely related to each other than to a third, and they're more closely related to that one than to a fourth, and so on and so on. And that's called a twin-nested hierarchy. So with this, and you can categorize all animals this way, and all plants and all fungi, and I mean organisms generally, but like if you figure out how to, if you can like do a phylogeny without it all getting screwed up by horizontal gene transfer, that's basically what you end up with.
Mm-hmm. Which makes it tough for some things like bacteria and archaea and things where there's a lot more horizontal gene transfer.
Right. And so for this one, they chose, they went with all animals, and then from there they picked chordata. And so chordata is a smaller subset of animals. It nests within total Animalia.
I'm a little bugged that all of the listed chordates are vertebrates. Come on.
Yep. Sorry.
Chordata.
No lancelets, no tunicates. Sorry.
Tunicates do not get enough respect. Oh, sorry for not having a brain as an adult. They still exist. Come on.
This is anti-tunicate propaganda.
It is. Although I will say that those pelagic tunicates need to learn their place and get back on the seafloor. Unacceptable.
Yeah. And then
I have strong opinion about tunicates.
I can tell. Yeah, you've told me about it before. You're just like, what are you doing? Sit down. Then within chordata, whichever, you have Mammalia, which is a smaller subset of chordata. So you start out with animals, and then we picked a subset of that, and now we have a subset of that, which is all mammals. And then we can pick a subset of that, which is carnivora, and a subset of that, which is canidae. Then Canis, which excludes the fox, which is vulpes, or the red fox is what I think that is.
I'm pretty sure it's vulpes.
Finally, Canis lupus, this one particular species within this genus. So what's cool about nested hierarchies is tunicates are not valid, says Mackenzie Brown. Well,
oh, yikes!
We get
fighting words around here, Mackenzie.
We have a lot of very strong tunicate opinions today. It's a sentence I didn't expect to utter.
It's a very divisive taxon.
So the cool thing about this nested hierarchy, which Linnaeus, a creationist, not an evolutionist, he was around before evolution was a thing, he figured this out. He figured you can nest groups within groups. So you have your total group, which has one set of characters, and then typically you have a smaller subset of that, which has all of the characters of that total group, all the characters which are ancestral to that total group, but also with their own characters on top of that. So animals are typically defined as multicellular eukaryotic phagotrophs. So they have true nuclei, they're multicellular, and they ingest their food.
They have to eat other organisms.
Yeah, they have to eat other organisms. They don't, with the exception of placozoans, they don't like excrete enzymes that break down their food like fungi do. Placozoans are just weird. We don't talk about them. So they eat their food, or they eat other organisms essentially. And so within that group, you have chordates. Chordates are still multicellular eukaryotic phagotrophs, but they've also added a postanal tail, pharyngeal arches, a dorsal hollow nerve cord, and a notochord.
At some point in their life cycle.
And then,
shout out to the tunicates.
On top of, at some point in their development, yes. And then on top of that, mammals have all of those characteristics, plus 3 middle ear bones, lactal mammaries, and synapomorphy for carnivora. Sorry, do you, Dapper?
So let's see. There are aspects of the skull involving things like dentition.
Mm-hmm.
Like they have a certain range of dental formulae. They have things about their inner ear. The shape of the orbital bars are also things, but like it's all like super technical.
Sure. Yeah. It's just like these minor skeletal bits, basically.
Yeah, like the difference between Canidae and Felidae like comes down to like differences of the skull, mostly. And you would think it might be retractile claws, but no, because retractile claws are actually ancestral to Carnivora, so you can't use that.
Yeah, foxes can do that, can't they? Can't they retract their claws?
I don't know about foxes being able to, but I do know that it was lost several times independently in Carnivora. Like, it was lost by hyenas independently from dogs, and then cheetahs also then independently lost it among the felids.
Gotcha.
Basically, retractile claws are nice, but if you're spending all your time doing like pursuit hunting, it's not worth it.
Right.
Because it reduces your maximum speed and your stamina, because it actually takes energy to hold the claws retracted. Because what that is basically just taking the last digit on the finger and raising it up off the ground, and that actually takes like that's effort. Anyway, sorry.
They are not retractable for foxes. My bad, folks.
Yeah, I didn't think so, but like maybe there's something I didn't know about fox.
So Carnivora includes your cats, your dogs, your bears, your weasels, your seals, all those guys. Those are all, of course. And I don't know if anyone's familiar with them, but like there are other cat relatives. I mean, everyone has heard of like meerkats and those guys, but there are also weirder things that most people aren't familiar with, like the fossa, which lives in Madagascar, and the binturong, and
I was going to add that if you didn't.
Oh well, there you go. And there are other like lesser known feliformes, but pandas.
Hyenas are more closely related to cats than dogs. Fun fact.
Yes.
They look like dogs because of convergent evolution, but if you take a look at their skeletons and also their anal glands, just fun stuff there, they're much more similar to feliformes than they are to caniformes.
Nice. So this pattern, this nested hierarchical pattern, is what we would expect under evolution. That's not to say you couldn't produce this under, say, intelligent design, but it is not an expected pattern under intelligent design. You could produce, in theory, any pattern or no pattern whatsoever under intelligent design. Fusa, sorry. My bad.
Yeah, it's spelled fossa, but it's pronounced fusa. It's weird.
My bad. But there's no reason to expect this pattern under intelligent design. So
yeah, actually, interestingly, Linnaeus himself was surprised to find this pattern because there's like nowadays you'll find intelligent design proponents saying, "Oh yeah, well we would expect a nested hierarchy," but the fact is that in known instances of design, nested hierarchies do not occur unless it's an intentional design goal, and it was not actually expected by people who didn't already know it was a thing, which means it wasn't expected.
Yeah.
The fact that you're now post hoc rationalizing this as an intelligent design advocate is fine, but you can't tell me that that's actually expected because it wasn't by anyone who didn't already know they had to account for it.
Right. Exactly. Yep. So we do expect this under evolution, not under intelligent design. You can rationalize it into there, but it was a prediction of, or it was a prediction that organisms should fall into nested hierarchies, given what we understand about evolution, with the exception of like horizontal gene transfer, which is a complicating process. So, all right, next slide, please. And then basically everything you already said. Evolution necessarily produces nested hierarchies because it is a process of descent with modification. Thus, if evolution is true, then we should be able to group organisms into nested hierarchies. Existing features are gradually modified for new functions. And then from chapter 13 of Origin of Species, again, you guys need to read this book. It's a good book. From the first dawn of life, all organic beings are found to resemble each other in descending degrees so that they can be classed in groups under groups. So basically the stuff we've already been saying.
Fun fact, just like when we have designed objects and they don't fall into objective nested hierarchies, when we have undesigned things that aren't biological but do have descent with modification, we still get nested hierarchies. And two very obvious examples are languages. We have language families, and you can see that there are sister languages and whatnot, but also ancient texts that were hand transcribed, because scribal errors tend to get passed down to the next copy of that text that's copied from the text with the error. So these are essentially analogous to mutations. And so you can actually also get textual families based on which scribal errors are transmitted. And by errors, they're not always actually mistakes. Sometimes they're like intentional alternate spellings or abbreviations, but those still count as changes to the original text that can be carried forward. And so, interestingly enough, the only reason things like, I don't know, biblical manuscripts, which a lot of anti-evolutionists are big fans of, the only reason we can actually classify them and then find out what the most likely original reading is because of things like nested hierarchy and the ability to like look at what is likely the primitive condition of something that has descent with modification.
Mm-hmm.
So yeah, it's not just organisms. Undesigned things that have descent with modification tend to always form nested hierarchies, whereas intentionally designed things only do it if that's a design goal. So for instance, when people construct languages, they don't fall into a nested hierarchy. Like there's no close relative of Klingon or Dothraki. However, some people intentionally make their languages to look like they come with a history, like Tolkien did with his Elvish languages. So because that was a design goal, there is a nested hierarchy. But he had to intend for that. It wasn't just an accident.
Right. Yeah, absolutely. It's good examples. Oh, that's right. That's what I was thinking of. So the example that say anti-evolutionists tend to give with, well, this skateboard is never going to evolve into an airplane. Well, yeah, but these do not fall into nested hierarchies, and they also don't reproduce, which is kind of the foundational thing for evolution to occur.
By the way, unlike languages and texts, which actually do reproduce in the fact of you get new speakers and you have new manuscripts copied.
Mm-hmm. Right.
Which is important even for those things.
Right, yeah. If it doesn't reproduce in any meaningful sense, like cars don't reproduce, there's no evolution. You can't make a nested hierarchy of cars because you can swap lots of car parts around, and there's no and because they don't reproduce, there's no genetic material. There's no way to determine which character is more weighted than any other characters, right? Lots of parts can be swapped out equally well, essentially. So anyway, so next slide, please. Another really good piece of evidence for evolution is morphological homologies. So again, this is sort of what we were talking about with the characters that are, within the common ancestor of a group, and then the different members within that group as they evolve and separate from each other will modify these structures in different ways. So in that picture on the left, you have those are different tree hoppers, and the structure which is highlighted by the little white dash, those are this is called the pronotum. It's right behind the head. And so in some you have this really elaborate pronotum, some you really don't. It's pretty small. But these are all it's the same structure, just modified in different ways over time, but they all have the same structures, right? And then on the right, you have vertebrate eyes. We talked about the evolution of the eye earlier, or sorry, in the previous episode. And so these all have the same structures, by and large. In some species like hawks, or some organisms like hawks, you have like a second fovea, or maybe some other added characters, but they all share the vertebrate photoreceptors and sclera and cornea and all that sort of stuff, right? So, anything you'd like to add, Dapper?
We should have had cephalopod eyes on here.
Well, that would be a convergence rather than a homology.
Not if you're just doing all cephalopods.
I mean, I guess that's true. But it's too late now. Anyway, next slide, please. More on morphological homologies. All tetrapods and even some lobe-finned fish have the same basic arm plan. So you have, birds, they've fused a bunch of their fingers. Lizards, lizard, rabbit, frog, they haven't really fused their fingers as much, at least in this picture. Then you even have the dolphin fin, which it's the same bones. I think there's hyperphalange. They've increased the number of finger bones they have, but besides that, all the other bones are the same. They have a a humerus and an ulna and radius, and then the carpals, metacarpals.
I'll also point out phalange count is actually pretty highly variable among tetrapods. So hyperphalanges that's not actually that unusual. It's weird for mammals. Mammals have a pretty strict 2, 3, 3, 3, 3, but
yeah,
even then, like, you can get supernumerary phalanges with relatively minor mutations, and so yeah.
So yeah, and then I have Anchiornis down there just to show you a close relative of birds that doesn't have the fused. The fingers aren't fused to the same extent, which is
they're actually
cool.
So
almost like
skeletally, they're not fused. It's unclear whether or not there was syndactyly between digits 2 and 3, which means that they would have shared like the same envelope of flesh. That's unclear, but they might have.
Gotcha. Yeah, but
actually, a lot of Maniraptora.
It's almost
there's. Oh, sorry. Go ahead.
It's like bird. I was just saying, it's almost like birds are theropods. They're just fused. What would you like? What were you saying?
Oh, I was just gonna say that.
Dapper.
Can you hear me?
Yeah, I can hear you now.
Okay, sorry. I might have cut out there. So a lot of Maniraptorans, there's actually some reason to think that maybe the digits 2 and 3 actually shared that envelope of skin, which is what tends to lend itself towards the fusion. And the reason for that is that you can actually see it in Anchiornis. They're in very close association skeletally, like they're directly parallel to each other, and it doesn't look like they could flex independently. So there's really, at that point, no selection pressure against mutations that would end up putting them into the same, like, big old clumpy finger. And then we know that that's the case for modern birds. So at some point, the first step towards that condition in birds is syndactyly. So at some point in the evolution of, Maniraptora into aves, which, birds are a subset of Maniraptora, syndactyly had to occur. And so there's a question is exactly how wide it was. I've seen people do like oviraptorosaurs with digits 2, 3 syndactyly. It's more common for dromaeosaurs in terms of reconstructions, but it's speculative no matter where you put it, but it had to occur somewhere. So that's just a fun little theropod thing.
Yeah, absolutely. And then another Darwin quote, because why not? Also from chapter 13. He said, "What can be more curious than that the hand of a man formed for grasping, that of a mole for digging, the leg of the horse, the paddle of the porpoise, and the wing of the bat should all be constructed on the same pattern and should include the same bones in the same relative positions." So essentially, there's no reason for us to have the same, bones as a mole and all the same positions and, the horse and everything, unless we all shared a common ancestor who also had that arrangement and then just modified it for different ends in different lineages. That is the most parsimonious explanation. It is decidedly less parsimonious to say that there was an intelligent designer who just used the same designs over and over and then modified them within different groups.
Yeah,
that's an extremely non-parsimonious conclusion.
In fact, you can actually even see places where animals have limb structures that don't really need to be the way they are. In fact, but actually be better not being that way. So, for instance, cattle have what's called a cannon bone, which is a fusion of all the metatarsals or metacarpals. They could just grow a single long bone there in embryo. That would be simpler and probably stronger, but they don't. They grow separate metacarpals or metatarsals, which then fuse into one big bone. Birds do something similar. They don't have to. They just do.
Right. They're I think, what is it, like part of their pelvis fuses, doesn't it, during development?
For birds, yeah. So that's actually a dinosaur thing. The sacral vertebrae fuse into the iliac crests and form one big thing. And also, characteristically, dinosaurs have more than 3 sacral vertebrae, and often up to like 13 or 14. Like, they go crazy with it.
Right. Yeah, and I think we're going to talk about embryology shortly, so I don't want to jump the gun on it. But yeah, there's a lot of stuff like that where it's like, why do we go through our, the development essentially if things were just designed this way and did not share common ancestry? Like,
why did dolphins all grow hind limbs just to reabsorb them?
Well, that's I think we're going to cover that one in a second. But there's another example. I think it's like the ossification of like the Meckel's cartilage. Like, there's no reason for it to go through this process unless that's how it occurred evolutionarily. Like, unless we had a common ancestor who did it this way. There's just no reason for that.
Mm-hmm.
Yeah, lots of organisms have parts that then fuse or are lost before they are born, and
like brains
why?
sometimes.
Yeah, like why? Yeah, why do tunicates need brains for part of their life cycle, but then not later? What's the point of having that in the first place? So anyway, and I think we'll talk about that in a second. So next slide, please. Vestigial structures. So as organisms adapt to their environments, sometimes certain structures can be reduced in size or in function, and typically by contrast, this enlarges the function of another structure. So for instance, vestigial means it has been reduced in size or function. It does not mean a structure is useless.
Nor has it ever.
Yeah. It just did not at any point mean that.
It's a common misconception, and it's sometimes perpetuated by actual textbooks, which is unfortunate.
Yes. Yeah. You can have researchers who will say, I mean, even if you want to define vestigial as useless, you can still find examples of useless structures in nature, and we'll talk about one in a second. So the forewings, which are not useless, they're in flies and Strepsiptera, they are used. The halteres, sorry, they are still used, but they are very, very reduced wings. They're used more like for maintaining stability. Not so much, but they're obviously not used for flight anymore.
Yeah, they're basically biological gyroscopes.
Gyroscopes, yeah, that was what I was looking for. But that's what they're used for now. They lost that original function in flight because they are obviously wings, or were wings. They're in the same place. They're they come from the same tissue, all that sort of stuff.
They're controlled by the same kind of muscles.
Right, yeah, but they're just not used as wings anymore. So they are vestigial. They lost that original function.
Also, Dipterans are the most deadly animals in the world to humans.
Yep. Everybody loves mosquitoes. Yeah.
There's very few organisms for which I advocate actual extermination, and some groups of Dipterans are in that group, in that category of things that I'm like, yeah, maybe humans should just make things extinct. Like botflies.
Yeah, botflies are pretty nasty. Those are.
We don't need botflies.
The book that I'm about finished with is oh crap! It's by Carl Zimmer. Parasite Rex. There we go. Parasite Rex. And it's all about. CRISPR says humans are the most deadly animal to humans. Yeah, that's probably true.
Well, actually, I don't know, man. I mean, malaria kills a lot of people. But I'm not convinced that more humans kill humans than malaria. Maybe though.
But so yeah, Parasite Rex is a really good book. It talks about.
Jackson, you're breaking up.
Did we lose Jackson?
Yeah, bot flies, nematodes, stuff that that gets in.
Yeah, Jackson, you're breaking up. We can't hear you.
Can you hear me now?
We can hear you now.
Currently, yes.
Okay, awesome. If it drops out again, just tell me. The ear muscles, the auriculars or auricularis muscles. So those are used in other.
They're used in other animals to help move the ear so that they can pinpoint the direction of sound. Whereas in humans, at most, they can just raise the ear like a few centimeters.
Or turning the ear towards sound.
Jackson, you might want to refresh the screen.
Can you hear me?
Yeah, you're dropping in and out. You might want to.
Okay, yeah, that's fine.
Refresh.
Alrighty, move to the next slide and let Dapper talk about that while I refresh.
I'll do that. That's fine. Okay, so atavistic structures are sort of like the inverse of vestigial structures. Vestigial structures are the remnants of ancestral structures that now have a reduced function, at least reduced at the original function. Vestigial structures are ancestral characteristics that are generally speaking lost in an organism, but which can reoccur. And this happens because basically the developmental pathways that would normally produce these in adults are still present, but they're usually just inactive during the embryological development. And so, for instance, on the left, we have polydactyly in horses. So horses, if you can see on the left, we have two bones listed as 2 and 4. Those are actually the metacarpals for digits 2 and 4. And these are called splints because they look like a splint that you would on someone's broken arm or leg or something, right? So these splint bones do not occur in all horses, but they actually occur in a fair portion of them. And they are metacarpals or metatarsals for digits 2 and 4, which generally don't actually form phalanges. But then in the middle picture and the one on the right, we have digit 2 reasserting itself as an actual present digit complete with a claw. That's very rare, but it does happen. Also, here we have hind limbs on a dolphin. So in embryo, all cetaceans grow hind limb buds, but generally speaking, they're absorbed back into the embryo before it even reaches, being formed. But in a handful of instances that have been documented, there were cetaceans that actually did have hind limbs. And so if horses were designed from the start to just have one hoof, then it seems unlikely that they would ever bother to get another one, because why would they even have the genes that would allow for the development of extra digits? Similarly for dolphins, if dolphins were just, separately arising from nothing else, no four-legged ancestors, and they don't need hind legs, and they typically don't have them, why bother developing them? So this is further examples because it seems unlikely if there weren't ancestral conditions for things like this that look like more broad or more basal conditions to reassert themselves in modern organisms. And Peter, could you move on to the next one? I'm not sure.
Can you guys hear me again?
Oh, I can hear you now.
Okay, that was a very good job.
Thank you.
I agree. Next slide, please. Yep, more on the embryology. As Dapper just mentioned, whales develop, or cetaceans develop hind limb buds that recede. On the other side, on the right, you have tunicate. This is tunicate larvae, which have notochords during development, which they then lose. Again, why would they need that if they're just going to lose it? What's the point? Well, the point is they are descended from ancestors who also had a notochord and dorsal hollow nerve cord and a brain, in their development. Would you like to add that?
Just that tunicates are valid unless they're free. That's it. That's all I got.
Fair enough. Next slide, please. So we also share much broader development with other organisms. So in that picture on the right, we share pharyngeal arches, notochord, dorsal hollow nerve cord, and a post-anal tail during ontogeny with all other chordates, all of them, everything from elephants to birds, to reptiles, amphibians, fish, tunicates, lancelets. We share these characters with all of them at some point in our development. Again, no reason why that should be the case unless we all came from a common ancestor who also has that, or that set of characters. And then on the left, you have, so we're deuterostomes. We develop our anus before our mouth embryonically. And we share that characteristic with all. Okay, we'll explain it, Mackenzie Brown. So deuterostomes, deutero means second, and stome means mouth. That means the mouth forms second. The anus forms first. Because when you are a gastrula, the first, or the digestive, or sorry, when you're a blastula, the blastopore becomes the anus. The first opening of your digestive tract becomes the anus. Whereas in protostomes, the blastopore becomes the mouth. And so you develop your mouth first. Proto, first, stome, mouth. So deuterostomes, that includes us. So all vertebrates, all chordates, plus echinoderms and hemichordates. So echinoderms are starfish, sea cucumbers, crinoids,
urchins.
sea urchins, all those guys are echinoderms. And then hemichordates are the acorn worms and graptolites, things we don't tend to.
You've probably
think
never
about
even
those.
heard of them.
But they're most people probably haven't. Graptolites are another a really cool index fossil. They're used to date different layers of rock, like phytoplankton. So really cool guys. Highly recommend checking them out. And then protostomes are pretty much all other bilaterally symmetrical animals.
Yeah, insects,
with like
mollusks.
the sole exception of xenocoelomorphs.
Yeah, it's like insects, mollusks, brachiopods. Brachiopods. I mean, like, let's see, velvet worms, like almost everything that you can think of. Arachnids, all arthropods, basically.
Yeah, the vast majority of bilaterally symmetrical animals are protostomes.
Yep.
Deuterostome is just a weird little side shoot.
But they're the biggest animals, so take that.
That's true. We are the biggest. So again, only makes sense, or that makes the most sense. It is most parsimonious to conclude that these guys shared a common ancestor who also had that developmental program, rather than it was independently bestowed on all of them for some reason.
Well,
because why not? I guess
there's a good case to be made for creationism. Because according to what you just said, all humans start out as assholes, and due to the fall, there's a decrease in information, so some of the humans will remain that way for the rest of their life. Good case.
Yep, I get that tracks. That checks out.
Evangelical Christianity 1, atheism 0.
Yep.
Next slide, please. Alrighty. This will probably be our last slide. So, fossil predictions.
No.
So these are fossils
4 more to
that
go.
We can do one more slide. Come on.
Is there only one slide after this?
No, 4.
Oh, 4? I think we can blaze through 4.
Sure, we can do that. That's fine. Okay, I didn't realize how many we had left. Okay, fossil predictions. So these are predictions. These are fossil organisms that were predicted specifically if evolution were true. Like if certain organisms shared a common ancestor, these organisms should exist, and they were found. So Microraptor, so a theropod, a non-avian theropod, with asymmetrical flight feathers on its front and back limbs, with partially fused arms, whatever, all that sort of stuff, should exist if birds are descended from non-avian theropod dinosaurs. Holy crap, there he is. This guy was predicted like a century before he was found.
Yeah, it was given the hypothetical name Tetrapteryx, which just means 4 wings.
Yeah. Diarthrognathes was predicted if mammals are descended from quote reptiles.
But we have Probanognathus up.
Probanognathus, sorry. Diarthrognathes, Probanognathus, they're in the same group.
Yeah.
Tridiolodonts or Trithelodonts, whichever one of those. But basically, you have a bunch of bones in your jaw if you're a reptile, quote. If you're an early reptile, and then for mammals, these other bones shifted up to become the inner ear, and basically the dentary became the only lower jawbone. And so, while these bones were moving up, there was a double connection that was happening between the dentary. Was it the dentary quadrate and the articular? Was it the articular squamosal?
So you have the articular quadrate connection, and then you have the dentary squamosal connection.
That's what it was, yeah. And so, this happened as a result of these bones moving upward. And again, there are no living organisms that span this gap. There are no living Probanognathans. So only if evolution is true, and these guys, and we share a common ancestor with reptiles, should this guy exist or have existed. And would you look at that? There he is. Sphica myrma is basically like an ant wasp predicted in the Cretaceous. It was predicted to have existed in the Cretaceous based on the diversity of Eocene ants, if I remember correctly. And E.O. Wilson and colleagues found it in Cretaceous amber, just like they expected they would.
Weird.
And then, of course, Tiktaalik. I think, have we talked about Tiktaalik in this series? I feel like we have, but
I'm not sure. But basically, there was the idea that, well, if we wanted to find the transition between sort of relatively basal, stegocephalian lobe-finned fish and tetrapods, you got to look at a certain period of time in swampy areas. And so an area that had an outcropping of the right time and the right environment was selected in far northern Canada, where no human should ever go, where I certainly will never go. But they did go there, as insane as that might be. And after years of digging, they did in fact find an organism that matched this description.
Yep.
And it has limb-like fins, and it has a head like, but not quite identical to early amphibian grade tetrapods. And it also has the beginnings of a neck, where the pectoral girdle is separated from the cranium, because in most vertebrates, the cranium and the pectoral girdle are the same complex of bone, whereas in tetrapods, they're separated.
Yep. Next slide, please. That's right, genetics. And so really fossils, homologies, nested hierarchies, those were all arguments that were sort of predate genetics, because genetics or genetic sequencing didn't really become a thing until the 70s. And, and then sort of got going in high gear starting in like the 80s, basically. And so with the advent of genetic sequencing, the evidence for evolution is essentially incontrovertible at this point. It'd be very, very, very difficult to explain the morphological and the fossil and the genetic data in terms of some non-evolutionary process for organisms. It would be very difficult.
The only option is intentional deception at this point.
Basically,
if something designed life to look like this, it was intentionally trying to deceive us into thinking that evolution had occurred.
Right. Yeah. There really is no other way to explain that. This is a quote from Proofer et al. 2012 when they sequenced the Ulindi genome, which is a bonobo. It said, on average, the two alleles in single copy autosomal regions in the bonobo genome are approximately 99.9% identical to each other, 99.6% identical to the corresponding sequences in the chimpanzee genome, and 98.7% identical to corresponding sequences in the human genome. Huh. Weird. Almost as if they're our sister genus. Crazy stuff.
Well, your sister genus.
Yeah, fair enough. So I think the chromosome they're looking at there are like, what are they called? P and Q or something like that. It's chromosome 2, essentially, because you have 2 for the others, for chimps, gorillas, and orangutans, who all have 24 pairs of chromosomes, whereas we only have 23. And the reason is a pair smashed into each other and fused end to end. Their telomeres fused with each other. And so now we have this really long chromosome, which is actually 2 chromosomes smashed together.
Complete with 2 centromeres, 3 telomere sections, including the telomere section that has a telomere-specific gene that is occasionally expressed, but just doesn't do anything, smack in the middle of human chromosome 2.
Yep, there's, again, intentional deception is the only way to explain that otherwise.
Basically, yeah.
There really is no other option at this point. Next slide, please. Okay, so this is where we're talking about endogenous retroviruses, which are really cool. So I don't have the actual structure of the virus up here. I apologize. But endogenous retroviruses essentially have 3 genes. So they have these long terminal repeats on either side, which are basically like repeat adenine or A and T rich. Or is it A and T or A and I think maybe it's A and G. A and G rich regions on like either side. Then you have essentially 3 genes, which are called GAG, POL, and INV, which code for different proteins, which help the ERV get transcribed and then put back into the genome in a new place, or recopied, etc. And so we have these ERVs. Well, there are types of these, and some just in humans.
I think we lost Jackson. So I'll step in here while Jackson is
like ERV 1,000 years old. But can you hear me again?
Yes, we can now.
I do apologize for that.
You want me to take this one while you try to get back?
Go ahead. I'll redo it. Go ahead.
Okay, so these ERVs, like Jackson said, are identifiable by the structure of the genome. And so the idea is that an ERV starts out as a virus, which will then insert its genome into the genome of another organism. And when that organism starts to transcribe its genome for making proteins, then the viral section will then be transcribed, and it will start to form new viral particles in the cell. However, sometimes these things get into the cell and then they're inactivated in certain ways. Maybe there's a mutation so they don't actually they're not actually expressed, or the various organisms have methods to isolate viral genomes that are mutations and try to prevent them from reproducing. If these infections enter the germline, which is how you're going to get gametes, they can be passed down because it's now part of the genome of that cell. And so there are ERVs, or elements derived from ERVs, that are common to successively smaller groups. So for instance, there's an ERV that is common to all mammals. Then there are some that are common to just monkeys. Then there are some that are common to just old world monkeys. Then there are some that are common to just apes, then just chimpanzees and humans, then just humans. And so because any given retrovirus is extremely unlikely to attack the same exact spot in the genome of two different organisms it infects, or even in just two cells that infects in the same organism, when you find these in the same place consistently across an entire taxon, it becomes virtually impossible to explain that without the idea that that taxon has a single common ancestor that was actually infected with this virus, which then reached fixity in the population. So the very fact that we have this nested hierarchy of just these essentially genetic parasites is extremely high evidence, extremely strong evidence for common ancestry among these groups that share these ERVs. Jackson, are we back?
Yes, I'm back. That's a
Okay.
very good explanation.
Thank you.
Other parts of the, so only about like 15% at max of our genome is actually involved in either making proteins or regulating the function of other genes. At least is what it seems like, which is a ridiculously small portion. And part of the reason is it turns out most of our genome are these, or what are called retro elements, and so basically they're pieces of our genome that get like copied and then duplicated. You know, they get copied, pasted over and over and over. And as a result, we have just copy after copy after copy of these different retro elements, like ALUs, for instance. 11% of our genome is just these repeats called ALUs, which are in all primates. Actually, I think they're in all euarchontic so rodents, rabbits, and tree shrews, colugos, and primates. We just have these sequences, and they're like partial ERVs, if I remember correctly. But you know, why do we need these things clogging up our genome if they're not really doing anything? And in fact, it turns out a lot of them are associated with different diseases, different types of like cancers and stuff. So that's fun. That's not to say all ERVs or all retro elements in totality don't do anything. Actually, some of them have been refitted for new purposes, but that's also not what the argument for evolution is. The argument is these unique inserts have a very, very low probability of being independently inserted into the same region of the genome. And so the fact that they are there in the same region for different species is indicative of common ancestry. That's the point. It has nothing to do with whether or not they're functional. And
by indicative of common ancestry, we mean like one of the strongest results that I've ever seen in an actual statistical analysis is indicating things like common ancestry given things like this. You're getting things like chances of the null hypothesis, separate ancestry, being true of something like 1 times 10 to the negative 119.
Yeah, something like that. It's bonkers.
Which is, it's so close to zero that I've never seen statistical results like this in anything. It's absurd.
Right. Yeah, absolutely. Next slide, please. That's right, paleogenomics. So we can also take these concepts of like generating phylogenies with genetics, and as it happens, some fossils are recent enough we can get genetic or proteomic data off of them and actually generate phylogenies. And turns out these organisms fit near other organisms. They're not just totally wacky separate creations or something like that. They fit within a nested hierarchy.
Wouldn't you know?
Wouldn't you know? So like Toxodon is that guy in the middle on the left side. Sorry, is it the guy on the top on the left side? Then Macrochenia, and then I think that's Paleoricteropus, which was like a weird Afrothere that was found in Madagascar, and it wasn't known what it was until the proteomic data came out. And it's also been done with like Glyptodons, which are armadillo relatives. I think Smilodon. And
Dons are like armadillos trying to be ankylosaurs.
Yeah, and like a few other guys. I think it's happened for some elephants and a couple human species, a couple ancient human species. So we're getting more and more and more data. This is a field that's only come into existence within like the past 20 years. This is a very new field. So,
which is really cool.
It is really cool. Anything you want to add?
Only that there's also attempts to do things like recover ancestral sequence in genomes, where like you're looking at things like taking into account paleogenetic sequences that you've acquired and modern sequences, and then saying, okay, well, what's the most parsimonious like ancestral sequence state for a particular gene or something? Mm-hmm. So there are even attempts to reconstruct the original version of a genome, sort of like the people reconstruct the ancestor languages of language families, like Proto-Indo-European. Mm-hmm. And so there's that analog that's happening in paleogenomics where there's an attempt to say, okay, well, what was like the ancestral state of this particular section of the genome for this whole taxon of animals? So yeah, that's also cool.
That is pretty darn cool. I agree. All right, I think you said there's one slide left. Right.
Yep. Last one.
Okay, what's the? Oh yeah, I remember this one. Okay. So this is one of my favorite experiments. This is a really cool experiment because there are some people who like to say things like, oh, you're just drawing lines on a page. Phylogeny doesn't really mean anything. Nope, that's just a lie. Mm-hmm. And here's how we know why that's just a lie. So what these researchers did was they took these T7 bacteriophages, which are bacteria viruses. And what they did was they grew them in the presence of a mutagen. So basically they induced higher mutation rates in these viruses, and they let them go for a time, then they split them, and they let them continue to let them grow for a time in the presence of this mutagen, and then split them again, and then did that again. They did it several times, and so what they produced was a nested hierarchy, because you had one population split, let them grow, split them again, let them grow, split them again. So you get this nested hierarchy. So what the researchers did was they sent these samples to another group of researchers. They didn't tell the researchers which group each of the virus samples was from. And they had the researchers sequence the endonuclease gene for each of the groups, each of the virus populations. And what they found was they did this with 5 groups. So all 5 of the phylogeny programs that were applied to this endonuclease data return the correct phylogeny and ancestor-descendant relationships. All 5 of them. So that means that this phylogeny, these lines on a page, were independently verified by a different team who did not know what the original data was. They only had the genes. So it is absolutely incorrect to say that phylogenies are just lines on a page. They can be verified. And if you disagree with that, well, you're totally welcome to put up your own counterexamples. So anyway.
Good luck.
Yep, good luck with that, folks. And I think that's the end of the PowerPoint. Right?
Well, congratulations. We did it. It only took 6-ish hours.
Yeah, just about 3 weeks. Yeah. So, as I said at the beginning, my intention for this was to essentially generate a series in which you have both the main lines of evidence for evolution as well as the overall process of evolution, and I think we covered that all reasonably well, at least the major bits. And so, there you go, internet. Have at it. Thank you, Dapper, for coming on and
You're welcome.
sharing your vast knowledge with us. And then thank you.
You're welcome.
And thank you, Peter, for hosting as always. Thank
Always
you,
a pleasure.
everybody in the audience. Well, it's always a pleasure to have you. So good job, folks. Thank you, everyone, for coming, and we will see you all next time.
Bye.
Bye.
Bye.